Moving from the United States to Israel is never easy, and the First Steps program helped me. They helped me with establishing my new lab, my dream lab in the biomedical engineering department. They helped with shipment costs and also with flight costs and even with housing.
The greatest asset of any university are its faculty members who inspire and lead our students so that they reach their maximum potential. We must keep up with other leading universities around the world. To do this, we aim to recruit 30 new faculty members every year to replace those who leave and to keep improving our student-to-faculty ratio.
For me, there was always the intent of coming back to Israel as an academic, taking all the experience I can to help me grow as a researcher back in Israel. The First Steps program, I see it as a very important part of the decision to come back, of course, but also enabling every researcher coming back to establish a lab that could succeed and that could have world-class output. And of course, you have to have that investment, initial investment in the lab to build it in order for that to happen.
Through this recruitment, we also strive to bring home to the Technion some of the brightest minds and professors who are currently at other top institutions in the world. This recruitment drive is probably the worthiest investment for us and for you. We must recruit these people and bring others back home in order to maintain our position as one of the top-ranking universities in the world and the most important technological institution in Israel.
I think it's very important to encourage scientists with the relevant and timely experience to come back to Israel and into Israeli academia. And that's what makes the First Step program crucial for the success of that process and to enable these scientists then to enhance the skills of all the people around them, so students and of course other scientists.
The First Steps program really helped me come back financially to the Technion. I know personally of a few friends in the United States or Europe where this type of program would really help them come back. And this program would also help the Technion in returning these great academics back home.
Good evening, ladies and gentlemen, and welcome to our eye-opening webinar, live from Haifa, with Professor Yotam Bar-On. I'm Professor Dori Derdikman, a faculty member at the Ruth and Bruce Rappaport Faculty of Medicine at the Technion. This event comes timely when the UK entered yet another strict lockdown. Israel vaccinated more than 1.3 million people, and the whole world is desperately fighting COVID-19 and its consequences. We cannot wait for a miracle. Scientists around the world in general, and in the Technion in particular, are working around the clock. With events like this, we aim to help you to know, learn and understand the science behind this virus and the battle against it. Our speaker tonight is a promising principal investigator and assistant professor of immunology at the Technion, which is a leading institution at the forefront of cutting-edge research on the disease. Yotam Bar-On will explain the interactions between our immune system and the virus with its mutations or variants, along with distinctions between different vaccine approaches. We know how highly vaccination has been anticipated worldwide. For this reason, we invite you to ask your questions in the Q&A box at the bottom of your page. We are also recording this webinar to be rewatched and distributed. Knowledge is an important asset. We want to share it with you. Thank you for the Technion UK for organizing this event. We hope you will find answers to your questions. Good night, good luck, and stay healthy.
Thank you, Dori. And first, I would like to thank Jofi Postain for organizing this event, and also for the Technion for allowing me to talk about this very important topic. What I will try to do today is to give you the scientific background for you to really understand the hurdles of developing an effective COVID-19 vaccine. I will also elaborate about the vaccines themselves, those that are currently in use and those that are in late phase clinical trial. These slides depict the history of pandemics. The most devastating pandemic occurred at the 14th century. This is the Black Death pandemic that caused the life of 200 million individuals. These 20 million individuals were half of the human population at that time. In 1918, a new strain of influenza virus emerged, causing a very serious pandemic. This is the Spanish flu pandemic that caused the life of 50 million individuals. This virus also infected 40% of the world population at that time. And it's important to understand about flu that every almost 10 years, there is a new strain of influenza that emerges, causing a serious pandemic. In the late 50s, the Asian flu, the late 60s, the Hong Kong flu, and even just recently, in 2009, the swine flu that was also very aggressive. And besides the flu pandemics, we are still dealing and battling with a very serious pandemic. This is AIDS. HIV, that you can see here, was discovered 40 years ago. And despite very intensive research effort, there is still no vaccine and no cure. And this leads me to the current pandemic, COVID-19. Unfortunately, the numbers here are no longer relevant because this pandemic is still ongoing. First, a little bit of terminology, because in the media, the terms are usually mixed, causing a lot of confusion. COVID-19, this is the disease or the pandemic. SARS-CoV-2, this is the scientific name of the virus that causes the disease. And although we were not really prepared for the COVID-19 pandemic, there were many warning signs that such pandemic can occur. Ralph Baric is a researcher at the University of North Carolina. And for many years, he tried to warn us about this danger. For example, this paper from 2015, what he did is something very interesting. He went to bats, he isolated corona strains from bats, and he showed in his lab that the specific coronavirus that he isolated can replicate in cells that are derived from the human respiratory system. A year later, 2016, he did something very similar. Again, he went to bats, isolated a new strain, which he called WIVI, and showed that this strain can replicate in the human respiratory system. Not only that, when he infected mice, he showed that this strain can lead to a very deadly disease. Another interesting paper was published at 2007 by a Chinese group. So the Chinese group saw the culture of eating exotic mammals in southern China, and they tried to warn us from the problem of this habit. And I will read exactly what is written in the highlighted text. The presence of large reservoir of SARS-CoV-like viruses in horseshoe bats, together with the culture of eating exotic mammals in southern China, is a time bomb. So this paper predicted exactly how the pandemic will occur, and again, 13 years before the pandemic. Pretty amazing. And in addition to that, there are also much clearer warning signs. This is simply the fact that we experienced corona pandemics not so long ago. In 2002, we suffered from SARS outbreak. SARS is a member of the coronavirus family. SARS passed from, again, everything starts with bats. SARS passed from bats into raccoon dog, and from the raccoon dog into human. SARS caused a very severe disease, but fortunately enough, the ability of the virus to spread globally was very limited. This is why the total death count was only around 800. 10 years later, another coronavirus called MERS, Middle East Respiratory Syndrome. Again, everything starts from the bats, passed into camels, and the camels infected humans, specifically in the Middle East. Very deadly virus that caused the life of around, again, only 800 individuals because it wasn't able to spread globally. Another very interesting fact about coronaviruses is that there are 4 strains of coronavirus that really coexist with us. These viruses passed from rodents into humans hundreds of years ago. And the reason they coexist with us is because they don't cause a very severe disease in humans. These are the common cold coronaviruses. All they cause is very mild symptoms like the common cold. And one question is, what is the difference between SARS, MERS, and SARS-CoV-2? If we look at the fatality rate of people that got infected with these viruses, the fatality rates are much higher in SARS and MERS compared to SARS-CoV-2, the current virus. But if we look at the ability of the virus to spread, which means the human-to-human transmission, here we see a huge advantage for SARS-CoV-2. And this is the main reason why the current virus was able to spread globally, causing a complete global shutdown, while SARS and MERS spread only locally. And this is not only because of biological traits of the virus, but also, and it might be counterintuitive, but because SARS and MERS cause such a severe disease, the people that got this virus immediately were hospitalized. This is why they had less interaction with other individuals and less room for the virus to pass from human to human. But a more interesting question is, what is the difference between these common cold coronaviruses and SARS-CoV-2? Again, both of them are in the same family, the coronavirus family. And if we look at the structure of these viruses, on the right side, you see the common cold coronaviruses, and in the left side, SARS-CoV-2, the current virus. The structure is similar. The genes and proteins are similar. So what is the difference? It's very interesting because we cannot point out to a specific gene that is expressed by the current virus and not by the common cold coronaviruses. And about that, I heard a very intriguing theory by David Baltimore. He's a researcher that knows a thing or two about viruses. He discovered the reverse transcriptase that allowed the HIV, for example, to replicate. He won the Nobel Prize for this discovery. And what he suggested, that the difference between these viruses is that common cold coronaviruses are very common. This is why we are usually infected with these viruses when we are in a young age. Therefore, the elderly population is already immune to those viruses, and their disease is not that severe because we have immune responses that can fight, and also immune memory that remember the infection, and we can fight the infection very quickly. However, SARS-CoV-2 caught us without any prior immunity, and the elderly population, we know now that these are the people that are in high risk for mortality. This is because there is no prior immunity to this virus. A little bit about the structure of SARS-CoV-2. SARS-CoV-2 is an RNA virus, which means that its genetic material is composed of RNA molecules and not DNA, like in our cells. The virus is covered with a lipid membrane. And by the way, this lipid membrane, the virus steals from our cells. Viruses cannot synthesize lipids. But the most important thing for this lecture is that on the membrane, the spike protein is expressed. The spike allows the virus to adhere the lung cells and infect them. As I mentioned, this is an RNA virus, but it has many unusual characteristics. For example, if we look at the genome size of different coronaviruses, you can see it in this graph. On the very top of the graph, we find the coronaviruses, which means that SARS-CoV-2 has a much larger genome than the rest of the coronavirus family. And this is important because it has such a big genome, it had to reduce the mutation rate. Because if it will keep the same mutation rate, for example, as HIV or influenza, which are much smaller, this will lead to what we call hypermutated virus, a virus that accumulates so many mutations that it cannot even infect cells. So the virus SARS-CoV-2 had to avoid this scenario. And what the virus developed is a proofreading machinery. This proofreading machinery, it's like an editing machine. Each time that a mutation is introduced, the virus knows to remove this mutation and put the correct nucleotide. But I said actually every time, but it's not true because this proofreading is not 100% efficient. And despite the proofreading, we still see accumulation of SARS-CoV-2 mutation. This is a major problem with SARS-CoV-2 because although the mutation rate is very low, the fact that the virus was able to spread globally, infecting many individuals, it had many opportunities to change the genome because the mutation occur during the viral replication. And this is best seen in this graph. This graph shows you the spike protein, okay, the most important protein of SARS-CoV-2 in terms of vaccines. And what this graph shows you are the different amino acids of the spike. And you see that somewhere in the world, for each position, there is a mutant that carry a different amino acid than the original SARS-CoV-2. This is something that we really need to monitor and take into account when we think about the vaccine efficacy. And the reason I focus on the ability of the virus to change the genome and to mutate is because we know that there is a clear reverse correlation between the ability of the virus to mutate and the ability of scientists to develop an effective vaccine. The best example is HIV. HIV is a highly diverse virus. It mutates very, very fast, 40 years of research and no vaccine. Influenza virus also mutate quite fast. There is a vaccine, but the vaccine is not a universal vaccine, which basically means that the vaccine is not able to completely eradicate the virus. And this is why each year we have to update the vaccine. These are not so optimistic examples, but if we look at measles, this is a very optimistic example because for measles, we have a very effective vaccine that almost completely eradicates the virus. And if we look at the mutation rate of measles, it's quite low. So now, of course, our focus is SARS-CoV-2. Where does SARS-CoV-2 stand in the scale of this mutation or the mutation rate? It's much closer to measles compared to HIV and influenza. Very good news for us and for the vaccines. But again, I have to remind you that despite the low mutation rate, the virus spread globally, so it had many opportunities to change and to mutate. So it's a balance between the two. And this leads me to talk about the COVID-19 vaccine development. most of the vaccine are trying to do something very simple, which is to elicit broadly antibodies against the spike. These antibodies neutralize the spike, and by neutralizing the spike, they prevent the infection. And I really want you to be able to understand the idea of generating neutralizing antibodies against the spike in more details, so you can really understand the mechanisms of the vaccines. So I would like to focus on the interaction of the spike with the lung cells. And this is exactly what we see here. On the left side, you see the coronavirus with the spike protein. And on the right side, you see the lung cell. The lung cells express a receptor called ACE2. So in order for the virus to infect, the spike must bind ACE2. This is necessary for the infection. It's like a lock and a key. Without that, there is no infection, and this is what we are trying to inhibit by neutralizing the spike. In order to elaborate on that, I would like to show you a short video. On the upper side of the slide, you see SARS-CoV-2 with the spike protein. In the lower part of the slide, these are the lung cells that the virus wants to infect now. So in order for infection to occur, the spike protein bind ACE2. And in this process, another thing is happening. I will show it again for you to be able to see that. Here. So step one, the head of the spike bind ACE2, but in addition, the spike is cleaved to two parts. And this is also done by a cellular protein called TMPRSS2. And why is that important? Because the removal of the head exposes the stem, the tail of the spike. And this tail is like a spring. Now that the cover is gone, the spring can do its function, and this is shown here. So the head of the spike is removed. Now the tail of the spike can stretch, bind the cell membrane, and fuse the cell membrane with the viral membrane, and now the virus can enter the cells. So that's a crucial step of the virus life cycle that we wish to inhibit by eliciting and neutralizing antibodies against the spike. But what needs to happen in the vaccinated individual in order for that to happen successfully? Step one, what we actually do when we vaccinate is we inject a foreign antigen. For simplicity, let's say that we inject the spike protein of SARS-CoV-2. The first thing that needs to happen is that lymphocytes that are called B cells will meet the spike protein. But this is not enough in order to generate antibodies. Second step, dendritic cells, these are also white blood cells. They engulf the protein, the spike, cut it to pieces, and present it on MHC class II molecules. And they present this protein or the peptide to T lymphocytes. These are helper cells. They help the B cell to generate antibodies. So now the third step is that the T cell that met the spike will interact with the B cell that met the same epitope exactly. Now the T cell will secrete a lot of cytokines that will support the activation of the B cell. Now the B cell will start secreting antibodies. Many of these antibodies will not be good enough. We are looking for specific antibodies that can neutralize the spike. But basically now we achieved what we wanted. By the way, this will not happen unless we add adjuvant. Adjuvant are materials that simply boost the immune response, okay? Usually in vaccination, aluminum salts are used, and they mainly work on step number 2 and boost it. So, but this is only one thing that we want to achieve by vaccination. Another thing is maybe even more important than generating the antibodies. We want to generate immune memory. We want to generate a memory B cells that will remember everything that just happened here, and next time that we'll encounter SARS-CoV-2, the immune response will be able to eliminate the virus. This is shown here in this graph. When we are first exposed to a pathogen, it could be bacteria or virus, the amount of antibodies that are generated are quite low, not enough. But if we are exposed again to the same pathogen, the amount of antibody that our immune system will produce is much higher. These antibodies are also better, and the time it takes to generate this high amount of antibodies is actually shorter. So now let's think about vaccine. In vaccines, the first exposure is the vaccine. Second exposure is when the vaccinated individual encounter SARS-CoV-2, and the amount of antibodies that will be generated will protect this vaccinated individual from infection. And this is all due to the memory B cells that we generated during and after the vaccination. Okay, and now I would like to elaborate specifically on the different approaches for a COVID-19 vaccine. The most classic approach is to synthesize the spike, mix it with adjuvant, and inject it to the vaccinated individual. And this is quite a good approach. The companies that are leading this approach are GSK and Sanofi, that really joined together in a collaborative manner to really boost things. And they had quite a delay because although they were able to elicit a lot of antibodies in younger adults, in the elderly population, they were not able to elicit enough antibodies. Another company that is using the exact same approach is Novavax. This is an American company that are currently in phase 3 clinical trials. Another classic approach is simply to use a dead virus. The advantage here is that you don't need a very advanced technique or technology. What you do here, you take the virus SARS-CoV-2, you inactivate it by heat or by chemicals, and you inject the dead virus. And this is actually what the Chinese are doing, and the vaccines that was developed by the Sinovac, the Chinese company Sinovac. But I would like to focus on the more innovative approaches. One of them is to use viral vectors. What we do here is we use viruses that don't do much harm when they infect us, like adenovirus. We even make them weaker in the lab. We modify them in the lab so they will not be able to replicate. And now these viral vectors, they express the gene of the spike. So the vector is used to really express the spike protein in the muscle cells. The companies that lead this approach are AstraZeneca together with Oxford and Johnson & Johnson. So I'm sure you all heard that AstraZeneca Oxford vaccine is now approved for use in the UK. Very good news. And they are using the exact, they are using this approach. They are using adenovirus for the injection. Johnson & Johnson is also very interesting because this is the only company that is testing whether we can inject the vaccine only one time instead of a prime and a boost. In addition, the vaccine that was developed in Russia used the same approach, and also the Israeli vaccine. But the focus of the next slide will be on the mRNA vaccine, because this is the vaccine that some of us already got this vaccine, and this is the most common vaccine right now. But in addition, it's also a fascinating approach that we were exposed to only now in the last 3 months. But behind that, there is many years of scientific work to develop such a vaccine. And the companies that leads this approach, I'm sure you all heard of, Moderna, Pfizer, and BioNTech. Okay, so for those of you that don't have any scientific background, I would like to explain what is it exactly mRNA. So we have genes in our DNA. The genes transcribe mRNA. The mRNA is simply a code. It's different nucleotides that can be recognized by a protein called ribosome. So this code goes into the ribosome in our cells, and the ribosome can translate this code into protein. So if we look at mRNA vaccines, instead of injecting the protein, the spike, we are injecting the mRNA that encodes the spike. And what are the advantages of using mRNA instead of the classical approach, which is to use a protein? One clear advantage is speed. It's much faster to produce large amount of mRNA in comparison with producing large amount of a protein. And this is why if you look at the race for vaccine, you can see that right from the start, Moderna, Pfizer, and BioNTech led the way because their ability to work faster. Another advantage is that there is no need for an adjuvant. Okay, it's a very clear advantage that I will not elaborate on. Another thing is that we mimic the viral infection very well, because also when we are infected with SARS-CoV-2, the virus, the mRNA of the virus is recognized by our ribosomes, and the viral protein are expressed in our cells. And this is exactly what is happening also in the Pfizer, BioNTech, and Moderna vaccines. So now we are witnessing a real revolution in vaccines, because now maybe all these years we were using an inferior method. Maybe mRNA can also solve problems that we were not able to solve, for example, vaccine to HIV or a universal vaccine against influenza. So now there are many studies that are trying to understand the advantage of using mRNA instead of proteins. For example, this study by Michaela Locci from UPenn, what she did is to inject mice with mRNA and inject another group of mice with protein and adjuvant. And what she showed is that all the responses that we talked about before were much better when the mice were injected with mRNA. More memory B cells, more antibodies, and more neutralizing antibodies. Very interesting. And now I would like to show you the results from the preclinical and clinical data of Moderna, Pfizer, and BioNTech. I want to show you this because I want you to judge by yourself the efficacy of the vaccine and the possible side effects. I will skip the mice data. I will start with the monkey studies. The monkeys received, and I will start with the Moderna experiment. What they did is to inject 2 doses of the vaccine in a 4-week interval. And 4 weeks after the prime dose, they infected the monkeys with SARS-CoV-2. This is something, of course, that we cannot do in human, and this is the importance of having a good animal model. Okay, so the first thing that they tested is whether we see neutralizing antibodies in the blood of the vaccinated monkeys. Because if we don't get antibodies after the vaccine will never work. By the way, in the next few graphs, the y-axis is very confusing. So on the top of each graph, I wrote exactly what they wanted to test. So they wanted to test the amount of neutralizing antibodies in the blood of the vaccinated monkeys. And you can see that when the monkeys were injected with 100 microgram, there is a very high level of neutralizing antibodies, a very positive result. And they also did a very important comparison. They compared the amount of antibodies in the vaccinated monkeys to the amount of antibodies that are generated in people that recovered from the disease, convalescent plasma. And you can see that the amount of antibody after vaccination is usually higher, even in comparison with the convalescent plasma. This is a very surprising and very positive result. Then they tested the ability of the antibodies to actually neutralize the virus. In very short, what they did here is to take the blood of the vaccinated monkeys, dilute it, and check how much we can dilute the blood and still neutralize the virus. And you can see that even if we dilute the blood quite a lot, we still get a very good neutralization response. Again, if we compare it to convalescent plasma, the neutralization ability after vaccination is even better than what is seen in people that recovered from the disease. This was all the Moderna experiments, but just to show you that in Pfizer, we get the exact same thing. Again, after the boost, there is high level of neutralizing antibodies that are usually higher than what is seen in people that recovered from the disease. But okay, so antibodies are necessary, but the big experiment was whether these monkeys are actually protected from infection. And you can see that the monkeys that received 100 microgram were completely protected from infection. 4 days after the infection, we don't see the virus in the lungs of the mice. One caveat here is that if you look at the monkeys that were untreated, they were simply infected without any immunization, you see that the disease in monkeys is quite mild. And 7 days after the infection, many of the monkeys recovered. And this is just to show you again, if we compare Moderna and Pfizer-BioNTech, we see that also in Pfizer-BioNTech, 6 days after the infection, in none of the monkeys we see any evidence of SARS-CoV-2 in the lungs. But I mentioned the caveat of using the monkeys, and it was of course important to do the clinical trials. And what the Moderna did is to give again 2 doses of the vaccine in a 4-week interval. And these individuals are now followed for there is a follow-up of 1 year, which was not completed yet. But let's see the results. Again, we are looking at the neutralizing antibodies. And you can see that in individuals that receive 100 microgram of the mRNA, after the first dose, we already see quite a lot of neutralizing antibodies. But look what happened after the second dose, after the boost. There is a high increase in the amount of neutralizing antibodies. Again, if we compare this to what is seen in individuals that recovered from the disease, we see that after vaccination, we usually get even more, on average, we get more antibodies than what we get during natural infection. This was Moderna. Pfizer. In Pfizer, I suggest to focus on 30 microgram because this is the dose that we are actually getting when we are being vaccinated. And you can see the exact same thing. After the boost, there is a sharp increase in the amount of the antibodies that are usually, the amount is higher than what is seen in people that recovered from the disease. But one important question is, what about the older individuals? Because this age group is at high risk of mortality or severe disease. So they wanted to see that the vaccine actually works in people that are older than 65 years old. And fortunately, what we see is that if we compare the amount of neutralizing antibodies in people that are more than 71 years old to people that are much younger, 18 to 55 years old, the level of antibody is basically similar. I say this is very surprising but encouraging result. And maybe this is because they use mRNA and not protein, but it's still too soon to really indicate if that's the reason. What about side effects? So there were many participants in this trial, and the side effects are very, very mild. What you see here in each part, they compare the placebo group to the group that actually received the vaccine. And you see that the side effects are minor. We're talking about fatigue, headache, or muscle pain, specifically at the site of the injection. So nothing very crucial. And lastly, I want to finish with a couple of slides that will discuss something that I think currently is the most important thing that we need to monitor. And this is the question of whether SARS-CoV-2 can develop resistance against the vaccine. Because if the virus will develop vaccine resistance, it will be very similar to what we see in influenza, that each year we will have to develop a new vaccine and we will have to learn to live with the virus because the vaccine will not be able to completely eradicate SARS-CoV-2. So this is the most important question right now. And basically, it's too soon to answer such question because we just have to wait and see. But if we look at SARS-CoV-2 reinfection, the ability of the virus to reinfect a person that already recovered from the infection, if we look at these numbers, the numbers are quite low, which is a very good indication because there are many similarities between infection and vaccination. And if the virus was able to evade the immune response after the first infection, probably it will also be able to do that after vaccination. But the fact that the numbers or the percentage is quite low is a very good indication that the virus cannot evade these immune responses. One variant is actually very interesting. This is the Indian variant that was able to reinfect a 28-year-old female, which means that maybe this variant is more capable of evading the immune response. So this variant that is now spreading in India is very important to monitor and focus on this variant. Of course, that you also heard about the UK variant. So I want to also elaborate a little bit about the UK variant. This virus doesn't have a single mutation. It actually has a cluster of mutations. All of them are in the spike protein. Some of them are very interesting. For example, the mutation 501 enable the virus to bind better to ACE2. And this is one of the reasons why the UK variant can spread very quickly in the human population. Another interesting mutation is actually a deletion of 2 amino acids at position 69 and 70 that allow this variant to escape convalescent plasma therapy. So it seems that the UK variant has some ability to evade immune responses, but again, it's still not clear whether the UK variant is resistant to the vaccine, but many labs are currently working on that, and I'm sure that in the next few days, we will have a more definitive answer. My personal opinion or prediction is that the UK variant will not be resistant to the vaccine because what we saw with different viruses, HIV or influenza, the viruses that are can evade neutralizing antibodies usually have a slower replication ability. And this variant is the opposite. It spread much more quickly. But this is just an hypothesis. We will have to wait a few days and see if this variant is actually resistant or sensitive to the vaccine. A very important question at this step of the pandemic. And with that, I would like to, I will be happy to answer any question. This is me, by the way, getting vaccinated yesterday, and I'm still alive, standing. I'm feeling very good, so no side effects. Okay, so I will read a few of the questions. Does delaying the Pfizer vaccine from 3 to 12 weeks affect its effectivity? And has no, I don't think that delaying the vaccine will affect the efficacy. What is important is to get the 2 doses, and this is something that you hear now a lot in the media. As you saw in the data, after the first dose, you get some level of antibodies, but not enough. So the protection will not be complete without the 2 doses. So this is definitely something that can affect the efficacy. So I think that, in my opinion, the idea of getting only 1 dose is not a very good idea, because then you also allow the virus more opportunities to evade the antibodies, because the antibodies are in subtherapeutic levels. Do the different blood groups have different defenses to COVID? Wow, that's a very good question, which I don't know the answer to. I'm sure there will be a lot of virological and immunological studies that will test these kind of things, like blood groups or HLA genotype. But currently, I did not hear about a study that tested this. Okay, I understand that after we have received the vaccine, we will still have to wear masks to stop infecting others. Is this correct? This is actually correct, and I want to explain what is the reason for that. It is still not clear if the vaccine can inhibit the replication of the virus in the upper respiratory system, for example, in the nose. Actually, Moderna, in the monkey studies, they showed that the vaccine was also able to inhibit viral replication in the upper respiratory system, which means that we will not be able to infect others. But because this is still not 100% clear, the FDA or the NIH advised people that got vaccinated to still wear masks, because this data is, again, not 100% clear. The smallpox vaccine was around for 200 years before the virus was eliminated through vaccination. The polio vaccine was around for 70 years before polio was eliminated. How long will it take to eliminate the virus, in your opinion? That's a fascinating question, and we don't know yet, because we don't know if the virus will be able to generate escape mutations. If it will be very hard, but we'll just have to wait and see. Why do so many viruses are derived from bats? That's a very good question. I don't know about other viruses. I know about coronaviruses. So bats, there are 8 types of different bats. Each of them can be infected with more than 10 strains of coronavirus. And what is interesting in bats is that they really, in bats, coronavirus do not cause a very severe disease. This is why they are like a reservoir of coronaviruses, because they don't die from the infection and they can spread it. And yeah, especially in cases like happened in southern China, when you know the story about the meat market. Are any more similar viruses expected? Very good question. I think that if we look in recent years, this is the 3rd corona pandemic that we experienced. We had SARS, we had MERS, and now COVID-19. So unfortunately, I think that more are coming. It's safe to assume that. Why do the spike connect with lungs protein only and not with other organs? That's a very good question. So just to focus on that question, the spike recognizes ACE2 on the lung cells. ACE2 is expressed also in other tissues like the placenta, the eye, and the heart. But you have to understand something, that SARS-CoV-2 is a respiratory virus, which means that in many cases, the virus doesn't reach the bloodstream. It accumulates in the lungs, and therefore it will not even reach the other organs. There was one blood paper that showed that only 8% of infected individuals, you see viremia, you see the virus in the blood. Okay, does the virus show signs of artificial engineering? So there was a theory that the virus SARS-CoV-2 was actually made by some lab, but this was proven to be not true, because and I'll save you the details, but when you make a virus in the lab, there are like small scars, you can say, on the genome of the virus that can be easily identified, and you don't see any evidence for that with SARS-CoV-2. Can you combine different vaccines using different way of producing immunity to get a broader effectiveness? In theory, that's actually a very good idea. So it was already shown in the lab that if you take only one neutralizing antibody against SARS-CoV-2, the virus, at least in vitro in the lab, can escape this antibody. So combining different approaches, different antibodies, is definitely a good idea. If older people cannot make T cells, how do these vaccines help them? It's not true that older people cannot make T cells. So I don't see that. Oh, how about the risk of ADE, antibody-dependent enhancement? That's obviously a scientist that asked this question. So ADE is a process where an antibody, instead of inhibiting the infection, actually boosts the infection. And many labs are currently testing whether ADE exists. Hopefully not, especially because if you look at the data, they are able to generate a very effective neutralizing antibodies, and I do not expect these very effective neutralizing antibodies to cause ADE, but again, many labs are currently testing these possibilities. What about children and children vaccination? That's also a good question. So one thing you need to understand is when Moderna and Pfizer did their large clinical trials, they did not test people under the age of 16. And this is why now these individuals are not getting vaccinated simply because they were not tested. Luckily enough, the disease in these younger adults is usually mild, but of course not all the time. So we just will have to wait and prioritize elderly population at this time. Someone who gets the vaccine, can they still transmit the virus? Yeah, that's again the same question that I got before. It all depends if the virus can still replicate, for example, in the nose. According to Moderna data in monkeys, the vaccine also inhibits this process, but it's still not clear to really indicate that. What are the ingredients in the vaccine that would trigger anaphylactic responses? Yeah, so when the vaccine was given in the UK, even before the FDA approval, 2 individuals got quite a severe allergic response. but it is still really not clear because it's such a small sample size what would be the cause for that. When I look at the ingredients of the vaccine, I really don't see any specific product that will lead to such a response. But what the FDA advised right now, that people that experience before an anaphylactic shock will not get the vaccine right now before it will be further evaluated. What are the potential complication, long-term side effects of this new mRNA vaccine? Yeah, so this is also a good question because not enough time has passed since the initial injection to really understand about long-term effect. But you have to understand that the mRNA is expressed to only a short time. After that, it is being translated to a protein. So from that moment on, this is exactly like any other vaccines that we took. So I don't see a reason for a long-term complication. If a person had the disease without knowing it, could vaccination have a detrimental impact upon individual health? Oh, okay, that's also, it's a good question. So in the original trials, people who have been sick before or were infected with SARS-CoV-2 were excluded. But that's not because it's risky to get the vaccine. This is because it would be very hard to analyze the results and to understand if the effect that they see is because of the infection or because of the vaccination. So in my opinion, I don't see any reason for a person who got infected to get vaccinated. Even this person will only benefit from the vaccination. It will have even higher it will elicit even higher antibody responses against the virus. What is the percentage of population need to be vaccinated for herd immunity? Around more than 60% is needed for herd immunity. But it also depends on the virus. For example, the UK variant that is now spreading more quickly might mean that we will need more people vaccinated in order to protect others. Which vaccine did you get, Professor? I got the Pfizer vaccine, but I didn't have much choice. That's the only vaccine currently available in Israel. But again, I feel quite good. So, why is the Moderna dose 100 and Pfizer is 30 microgram? Yeah, that's a great question that I don't know the answer. So they use the exact same technology. In Moderna, we are injected with 100 microgram. In Pfizer vaccine, we get a lesser amount, 30 microgram. It's very hard to know why. I guess there are small differences between these companies. Some of them are kept, I think, as a secret because of patent issues. So I don't have a good answer for that. Have you heard about a drug against COVID being developed by Vital Brazil Institute, which uses the plasma of horses? So I haven't heard about this specific one, but the idea of using plasma is very similar to an active vaccine. What we are trying to do, instead of eliciting the antibodies by a vaccine, we are simply infusing the neutralizing antibodies. And there are many works now, different labs are trying to develop or already developed very potent neutralizing antibodies that will replace the plasma therapy, which means that you will inject a single or a combination of antibodies instead of injecting plasma. The advantage here is that you can use it for mass production. Whether plasma it's from one patient to the other, in the lab you can produce a lot of antibodies and inject it to a bigger population. More questions? How many COVID vaccines to date have demonstrated capacity to induce production of IgA antibodies? A very good question. So this, according to the trial, they tested IgG. And there was one paper or even more that showed that IgA would be more protective, and it makes sense because this is a respiratory virus. But I guess because the amount of IgG is so high, they are also able to penetrate to the respiratory system. Okay, the protocol for COVID-19 vaccine excluded people who already proven that they got the infection. What happened if someone already had COVID-19 infection, recovered, then got the vaccine? So nothing will happen. They were excluded from the trial because it will be extremely hard to analyze the data and to know if the efficacy is due to the infection or the vaccine. But if someone got infected and then got vaccinated, this person will only benefit from the vaccination. Okay, I think we ran out of time. Thank you all for listening. And again, thank you, the Technion, for giving me the opportunity to speak about this very important topic.